Assalamualaikum and greeting. In this video, we are presenting about hatchery management practical.
Fish held in groups of 10 to 12 in low salinity water of 3 to 5 parts per thousand. Tanks can reach to 26 degrees Celsius in summer. Tanks are covered in netting to prevent fish from jumping out. Tanks are kept free of solid waste with a broken siphon system. An angled PVC pipe draws up waste from of the tank for a tank overflow. The tank can also be brushed, cleaned, then flushed through a central stand part system. This system can be activated by opening a valve at the base of the tank. The incubator rooms at each facility are designed to mimic a natural flow environment. There is a constant flow of fresh water to supply oxygen and clean up waste. The incubator tray is filled with small plastic saddle-shaped pieces that serve as artificial pebbles called substrates. The substrates also provides a hiding place where hatching young salmon alevin can remain undisturbed until they have absorbed their egg yolk material for body development. Alevin is very sensitive to light, so the incubation area remains as dark as possible.
Most tilapia species breed in saucer-shaped nest built by the male at the pond bottom. The male build it for attract female to spawn inside the nest. Then the female spawning the eggs inside the nest. After that, the male fertilizes the eggs. Then the female takes the egg into her mouth and she swims away with the eggs for approximately 10 days until the larvae are developed. That's the moment where we collect the eggs from the female and put it into incubation jars. When collecting the eggs, we keep the different developmental stages of the eggs separately, so all the eggs in one jar are more or less of the same age. These incubation jars are where they stay in motion, just like when the egg is inside the female mouth. The larvae develop in this jar for about 10 days until they are fully developed. Then the developed larvae can swim up and go all the way up into a flowering tank where we can start feeding the larvae.
Artemia is a genus of aquatic crustaceans, also known as brine shrimp. The cyst outer membrane cracks, and the embryo emerge after 20-hour interval. Once completely emerged from the cyst, the nauplius begins a series of beating movements, which rupture the hatching membrane, allowing the nauplius to swim freely. The first larval stage is characterized by a distinct brownish orange color, a red nauplius eye in the head region, and 3 pairs of appendages. After 8 to 12 hours, it molts into second larval stage. At this stage, algae bacteria are filtered and ingested into the digestive tract by the animal. The fact of the Artemia, it capable of reproducing 300 new nauplius every 4 days. As they grow and develop, they undergo various stages, and each stage is separated from the nest by a mold. And here is an adult Artemia. Next, microworms are small nematode worm that can be used as food for small fishes. The lifespan of the microworms are 20 to 30 days, and it starts to reproduce at 3 days old and bear 40 young every 1 to 2 days, making them a very prolific breeder.
Larvae care and water management. First is larvae care. Shield the eggs. Shield the eggs from direct sunlight is important to avoid damage by ultraviolet radiations, especially during early development. Concentrations of dissolved oxygen. Concentrations of DO for artificial eggs incubation is extremely important and more effective than the churning actions of the female. Temperature. The temperature maintained the eggs incubation to serve the purpose. Water flow. Water flow into the incubators is really important to remove harmful metabolites such as carbon dioxide and ammonia. Next is water management. First is improved water quality is really important by using proper filtrations, chlorinations, or sterilizations. If this not be done, it will lead to mortalities, lead molding, and deformities. Water quality will be poor if reduced stocking density not be done. Increased water exchange. It will lead to mortality if water exchange not be done frequently. Improve tank and water disinfection protocol by using zero green vibrocollis and low level of yellow vibros on TCBS agar plates. It can reduce the bacteria in the water.
In conclusion, we know there are constant upwelling flow of fresh water, also how the chronology of breeding the fish. We also know that the artemia and microworms both are live food. And last but not least, taking care of larvae is a necessary. So that is all from us. Thank you.